be able to answer to the question, what are the enzymes? Enzymes are catalysts. A catalyst is a substance, meaning speed up the reaction, that initiates or accelerates a biochemical reaction without itself being affected. Now that you know what the enzymes are. Now let's discuss the structures of the enzyme. The activity of the enzyme determined by three-dimensional structure. The substrate molecule binds to its enzyme like a key and a lock. The enzymes are so specific that the self of the enzyme made in a cell determine which metabolic pathway occur in your cell. For an example, let's think of this. The large red is the enzyme, and these two orange are substrates. The enzyme will only bind to its specific substrate and substrate that fits to its structure. Okay, the area where substrate bind we called active site. As you see, this is the active site of the enzyme. If you can see clearly, this is the black line of the enzyme. This is the active site of the enzyme where the substrate can bind. Okay, as we see, the enzyme will not bind to this white parallelogram. Why? Because the structure is not fit. Okay, and when enzyme and the substrate binds together, the bond can form. Okay, now let's think that this tape represents the bond in our example. Okay, in our example, the tape represents to the bond, and the two substrates before create a new molecules that we call product, and the enzyme can do this over and over again until final product had been formed. As we said, enzyme structure determine function. So now let's talk about the role of the enzyme. After we have talking about the structures of the enzyme, now let us discuss more about enzyme and what is the important role of the enzyme in our body. Okay, the enzyme plays an important role in our body. They are used in the metabolic pathway throughout the body. Enzymes do this binding to one or more reactants of the reaction that catalyze. Enzymatic reactions are much faster than non-enzymatic reactions. This is very good because it alone helps your body to react or make an adaptation very quickly to the situation. So if you need something made in your body, it can make a reaction which happens very quick. Yep. In order to understand about the reaction, how enzymes catalyze the reaction, the energy of activation is needed. Have you ever heard about the energy of activation? Let's talk about this. Each reaction has a certain energy cost that it needs in order for that reaction to occur. So enzymes do this. It's to give you a discount low cost in the reaction to occur. It makes the reaction happen quickly. Assume the reaction is like this. This is the coordinate of the reaction. This is a reaction coordinate, and this is the enthalpy. Okay. One reaction is occur, but it's without the enzyme. This is without the enzyme. What the enzyme do is, when the enzyme come in, the enzyme make, this is the enzyme, assume this is the enzyme, okay, it comes in and make the reaction go fast. and it gives energy activation becomes low. Assume that this energy of activation is 100 cost. An enzyme can give you a discount, 50 activation energy. So the enzyme will come in and knock it down to 50 energy points. And I will give you one example of reaction that occur in our liver, isn't in our body. Let's talk about the catalyzed enzyme. Have you ever heard catalyzed enzyme? Catalyzed enzyme can be found in our liver, which is catalyze a reaction. Hydrogen peroxide, and catalyze, and this reaction is catalyzed by catalase, and forming water and oxygen in our body, because hydrogen peroxide is a toxic in our body. So this is the example of an enzyme in our body. Okay, the last but not at the least, you have understood what enzymes are, and you have known the structures of the enzyme, and also you have known the role of enzymes. Now let's talk about the factors that affect the rate of enzyme reaction. Now I will talk 4 of them. Okay, the first 2 are the temperature and the pH. If the temperature of the enzyme is really high, so the enzyme will denature. Quite high pH has the same effect. And the second 2 are the concentration of enzyme and the concentration of the substrate. The concentration of the enzyme and substrate affect the rate of the reaction. So that's all that I have about the enzyme. I hope this information is useful for you. See you again. Bye.
Enzymes are proteins which catalyze metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed, leaving a hydroxide ion. Carbon dioxide binds to the hydroxide ion on the zinc, and the product is released. When a similar shaped molecule competes for the active site, it reduces substrate activity. This is called competitive inhibition. Adding more substrate increases activity. In non-competitive, allosteric inhibition, a molecule binds to a remote part of the protein, preventing substrate binding by altering the shape of the protein.